I welcome you to episode 3 of our series of nucleic acids. In the previous episode, we were able to look at the DNA structure. And as you know, DNA structure must undergo replication. Why? Whenever a cell is dividing, the new cell which is being formed must look exactly like the parent. And therefore, for it to look exactly like the parent, the DNA which controls whatever was happening in the parent must also be present in the daughter cell. And therefore, that's why the DNA must also form a copy of itself, such that by the time this cell divides, the daughter cell which is being formed must have a copy of this DNA which was controlling those same things in the parent. And that's what makes us to come here to learn how replication takes place. Replication simply means a process by which a given structure forms an exact copy of itself. We are saying replication is a process by which a structure forms an exact copy of itself. So it means when we talk about DNA replication, we are trying to see how DNA forms a copy of itself, and I've already given you a reason as to why DNA must form a copy of itself. Another thing is, in the previous video I talked about a situation where we resemble people in our family. Now, it means that, for example, if it's a man and this man is going to form sperms, these sperms must get a copy of the DNA of that man himself. Why? Because these sperms must get a copy of this DNA. Likewise, if it's a woman, the ova of the woman must also have a copy of the DNA, such that when the sperm with a copy of the DNA of the father comes and meets with the egg, which has the copy of the DNA of the what? Of the mother, they can now be able to form a resultant product, which is me and you. So, me and you who are watching and who is presenting to you, we have a copy in our cells. We have got a copy of the DNA from our father and a copy of the DNA from our mothers, both in us. That's why, in case my father doubts me whether he is my son, whether I am his son, they usually go for DNA test as a confirmatory test. Why? Because within my body I should have a copy of his DNA. So what happens is they get my DNA and also they get the DNA of my father, and they start looking at how the bases follow each other. There must be a perfect match between how bases follow each other on my DNA, how the adenine, guanine, cytosine, how they follow each other on my DNA and the DNA of my father. There should be a complementary copy. My DNA should be a complementary copy. One of my strands should be a complementary copy of the DNA of my father. That's how they usually ensure that they check for paternity. Now, I know somebody can also still ask themselves that why is it that maybe somebody find themselves resembling more of their father and more of their mother. Still, the question is, yes, this DNA contains bases, but those bases, those A, G, C, T, A, R, A, E, those A, G, C, T, the way A, C, C, A, G, C, T, A. Now, how these bases are following themselves, how they are following themselves on the what? On the DNA, if this is a DNA strand. How these bases flow, this flow of bases, as we are going to be seeing in the next episode. That's why you need to make sure to subscribe. if you are not subscribed to this YouTube channel so that you can be updated on the next episodes we are going to put. We are going to find out that this based the way they follow themselves here, actually, there is a code they are giving. It's a secret code for a given gene, for a given protein, for manufacturing a given protein. Remember, anything which controls the protein, anything that controls the protein, that controls how a protein is made, actually controls whatever happens in you. Why? Because proteins are enzymes. Enzymes control almost everything. Proteins form hormones. Hormones control different metabolism and so on. So the DNA has got these codes. These codes are actually indirectly, they are genes. So it means the copy of the DNA that I got from my father had some genes. The copy of the DNA I got from my mother had some genes. So when the copy of the DNA of my father came and joined with the copy of the DNA from my mother and formed me, the offspring. Now, we go into other biology on which of the genes either are dominant over the other, are recessive over the other, which ones are codominant. In some cases, you find maybe some genes are codominant. Others, you find one, some of the genes maybe from my father are dominant over the ones from my mother. So in that case, you find myself resembling more of my father than less of my mother. And then in some cases, you find that some people, like for example me, I resemble both. When I'm moving with my mother, they think that actually I am more of my mother. When I'm moving with my father, they think that I'm more of my father. So in such case, it means in some cases, some characteristics blend, and you find you form something which is an intermediate. Not the 100% the mother, not 100% the father. I don't know you who is watching who I belong amongst those categories. But now there depends on of the genes which are the DNA that I got from my father and the DNA which I got from my mother. Which of the genes are dominant over the other? The one which are dominant over the other will make me resemble more of that. If the ones for my father are dominant, then I will resemble more of my father, or vice versa. So, we want to look at DNA replication. So that's why DNA must replicate. Why? The DNA must replicate so that the new cell that is formed, for example, during mitosis, the new cell which is formed is the exact copy of the previous cell, of the parent cell. Another thing, DNA must replicate, for example, during formation of gametes, during formation of sperms, such that genes which are found on this DNA can be transferred from one parent, from one generation to another. Why? Because the genes were transferred on the DNA that I got that I got from the sperms of my father, and then they are now into me. So that was from the generation of my father to my generation. Then likewise, also from me, the genes are going to be I transferred my genes to my children, like that. So that's how so it is from DNA replication that enables us, that enables genes to be transferred, especially from meiosis, which is formation of gametes. It enables genes to be transferred from the parent organism to the offspring, from one generation to another. That's why there is need for replication. I hope you have now seen the significance of replication. Now, for DNA to replicate, there are requirements which are needed. What are those requirements? For DNA to replicate, what are the requirements? Requirements for DNA replication. For DNA to replicate, what do you need? Remember, we have said replication is a full process by which a structure, like for now in this case, DNA forms a copy of itself. So it means we need the DNA itself. We need the DNA. It means we need a DNA original strand. We need original DNA structure, the DNA molecule. It means we need the DNA original molecule. Why? Because we cannot form a copy of something we don't have. So we must have the DNA original structure of DNA to form a copy of itself, of that DNA. So we need DNA molecule. Number 2, what do we need for DNA replication? We need energy. We need energy. Energy, this energy is in the form of what? This is energy in the form of ATP. We need energy. Why? the process of formation of the replication process is an energy requiring process, and therefore we must have energy reserves that we can use to have this DNA being formed. And as we are also going to see, we also need nitrogenous bases. We also need nitrogenous bases. Yeah, we need nitrogenous bases. As we shall see, we shall find out we need these bases. We need these bases because they are the ones which are going to help us to form another copy. We need these bases which are going to help us to form another copy. Why? As we shall see, we shall find out in some cases the original copy usually opens up, and when it opens up, the original DNA unwinds. When it unwinds, then you find ourselves having nucleotides. Nucleotides come to join up. Now, actually, in this case, I may not say we need nitrogenous bases, but I can say we need nucleotides. And we need nucleotides because it's not just only bases, but and we cannot have bases just free moving in air. We need nucleotides because these nucleotides are the ones which are going to come and join together and help us form a new structure of DNA, which is the exact copy of the parent, as we shall see. Just keep watching. And then what we need else is also we need enzymes. We need enzymes. As you know, that every process in our bodies is controlled by enzymes. Likewise, this process also needs enzymes. And these enzymes include, for example, we are going to find an enzyme called DNA helicase. We are going to see enzymes like DNA polymerase. We are going to meet this enzyme like called DNA polymerase. We are going to meet an enzyme called DNA ligase, and so on and so forth. So we need more of this, and we need these enzymes to also be present so that they can speed up the rate at which this process takes place. So with this present, then we can have DNA take place. We can have DNA replication take place. And here this replication process usually takes place it usually occurs during a stage of cell division, which is known as interphase. During interphase. Here in interphase stage, this interphase stage, that is usually that is the stage where a cell is preparing to what? Where the cell is preparing to divide. And in that process, as it is preparing to divide, very many things occur. For example, we have formation of reserves of energy, we have DNA replication, we have replication of different organelles, and so on and so forth. Yeah, it should takes place in that stage. So with all these things present, we are saying the cell now can be able to undergo, the DNA can be able to undergo replication. Now, we have got different types of replication. DNA replication. We have got different ways how replication takes place, and these different ways have been suggested. These different ways of DNA replication have been suggested by different people and what you call hypotheses. So we have got different hypotheses which have been put forward to explain how DNA replicates. And of course, among these ones, of course, we have the one which is most acceptable and one which is less acceptable. And now, what are these different types of DNA replication? Now, if I have my original DNA strand, assume that I have my original DNA strand as this. If I have my DNA original strand as this one here, I have my DNA strand, original strand, which is like that. Now, if this is the original strand which is present, the DNA, not strand, but the DNA molecule. This is my original DNA molecule like that. Original DNA molecule of DNA, as we have already seen, I have DNA molecule which is like that, as we have seen in the previous episode. Now, in one of the hypotheses that explains how DNA replicates, it says that this DNA molecule, which is like this, usually opens up. It unwinds, because when you see here, this strand and this strand, they are joined together. So in this case, one of the theories, one of the hypotheses is called semi-conservative. Is what you call semi-conservative. Semi-conservative what? Semi-conservative hypothesis. Now, in this semi-conservative hypothesis, it says that the DNA structure has to open up. It unwinds, it unzips. If this one was a zip, these original bonds were zips, which are zipping these two strands together. It unzips, and when it unzips, you have two strands which are just open. Now, those two strands which are just open, now each one of them acts as a template. Each one of them acts as a template for the formation of a new nucleotide, for the formation of a new strand. How? You originally had something like this. See? So when these hydrogen bonds are broken, and the strand or the double strand is unzipped or is exposed, what happens? You end up having two strands. You end up having one strand like that, another strand like that. Then now these strands which have now opened up will act as you have two strands which are like that. And you have these strands which have opened up like that. You have a strand which has opened up like that. Now these ones have opened up like that. The hydrogen bonds have been broken down. Now when they have opened up like that, now we have nucleotides. These ones I told you about, these nucleotides coming and they start joining up. Of course, following the complementary base pairing, what I've already explained to you in a previous video. So these nucleotides come and join up under what you call the complementary base pairing rule. And when they join up, you now end up, of course, these are bases which are exposed. These bases, they have got nucleotides which have got bases that are complementary to them. What am I saying? I'm saying these are bases which have been exposed. Now within the nucleus of a cell, we have nucleotides, free nucleotides. These nucleotides I've told you about. These nucleotides have got a base on them, of course. Now these bases, those nucleotides which have got bases that are complementary to these bases which have been exposed, they come and bond. And when they bond, of course, later they form phosphodiester bond between themselves, and then they end up forming a long molecule besides the original. So in that case, you end up having something like this. You end up having another new molecule which is coming and joining up like that. I don't know if it is. You have another molecule which is coming up like that. I don't know. So you end up having a molecule which originally was that, and then this one opened up. After opening up, a new nucleotides come and join up. When they come and join up, you end up forming a molecule which is like that. So when you check here, it means that you end up having, this was the original strand which opened up. It was the original strand which was opened up like that. It was the original strand which was like that. But after opening up, nucleotides came and joined together, and after joining together, they bonded with these nucleotides following the complementary base pairing rule, and then after they formed a new strand besides. So you also have another strand joined here, have another strand, then the two come and join together like that. They end up joining together like that, and you also form another structure there, another structure like that. So in that case, it means you have got, that's what you call semi-conservative. Semi-conservative, why? Because we are saying that one of the strand is the original strand, that's we have conserved it. But it is semi because the cells which have been formed, one of the strands is the original strand, that which we have conserved. Then another strand is brand new. So that kind of replication where the DNA molecule divides from two other daughter molecules, whereby each of the daughter molecules has or contains one of the strand which is the original copy, and then the other strand which is the brand new copy, is what you call semi-conservative hypothesis or semi-conservative replication. Then we have also got another one. We also got another type of replication where you have the original, but now this original remains when it is intact. It remains when it is intact, and then you form a new. you form a brand new strand, a new brand molecule, a new brand DNA strand like that, DNA molecule like that. So in this replication here, the original strand is there, but without tampering with it in any way. What happens is that a new strand is formed just besides the original, without the original being in either way tampered with. That kind of replication, we call it conservative replication. Conservative replication. Or it's also what we can also call we can also call it conservative hypothesis. See, that is mentioned. It is described under what you call conservative hypothesis, or you can just call it conservative replication. Here we are saying in this type of replication, the original strand is untampered with, but a new strand is formed just beside the original strand, which is the exact copy of the original strand. Are you getting what I'm saying? The original strand is untampered with, but through some way, a new strand is formed that exactly resembles the original strand. That's what we call the conservative. So it means here what we are forming here is called conservative, because you end up forming the original one has been conserved. We have not tampered with it fully. That's why I call it conservative. The original one remains original, untampered with. Then you form a brand new. But here, you are opening the original, you get two strands, then you use each strand as a template to manufacture a new one. So you ended up, for the semi-conservative, you ended up forming two daughter DNA molecules, but each of the two daughter DNA molecules, one has the original strand and a brand new. But here, in conservative, you conserve the original, you don't tamper with it, but in some way, you form another new besides that, and that's what we call conservative replication. Then we also have another one. Which we call dispersive replication. In dispersive replication, how is it? In dispersive replication, or what we also call fragmentation, fragmentation replication, or fragmentation hypothesis. What happens here is that you have the original strand. Now you want to form a new copy. What happens is that here it is just some segments, some fragments, or some sections of the original which are replaced with the brand new nucleotides. How? If I can do some illustration here, it is like this. You have the original strand, but here segments of the original strand are replaced by complementary brand new nucleotides. So it means in this case, this original nucleotide opens up, but in some few places, in some specific areas. And what happens is you end up forming a new strand which is going to be like this. A new strand which is going to be like that. I want you to look at what I'm meaning here. You'll be having a new strand which will be like that, then like that, then like that. Then you have something also like this, like that, like that, like that. So when you check here, I hope you can see a difference. I pray that you're able to see the difference here. When you check on this one here, what do you see? When you check on this one, I hope you are able to see a difference. How? In this one here, you see this one here is brand new. then this section is the original strand. Then another part is going to be is resembling like a brand new. Then another, if we were to continue more and more the other side, so you will be having a molecule whereby some sections have a brand new strands. The strands are brand new, nucleotides are all brand new. Then some sections of the same DNA, you have got them having the original structure of the DNA strand. That is different from semi-conservative, because in semi-conservative, a whole strand is brand new, as the other whole strand is the original DNA structure. The original DNA is from the original DNA molecule. But here we are saying fragments of the daughter DNA molecule. Here in this passive replication, the fragments, segments, or parts or fragments within the brand new within the new DNA molecule, some fragments or some sections or some parts have brand new nucleotides, while some parts have sections of the original DNA molecule. I don't know if you get me. Here in this passive replication, we are saying the daughter DNA molecule that is formed has some sections which are brand new and some sections which resemble the original DNA strand, as you see in this illustration here. Now, these are the different ways how DNA undergoes replication. But now, of these three hypotheses, the semi-conservative hypothesis, conservative hypothesis, and dispersive hypothesis, which one is more reliable, or which one can more acceptable? Of these ones, we are going to rely more on the semi-conservative hypothesis. This one, which I've already explained to you. So, in the next episode, we are going to look at this semi-conservative hypothesis in details. And in case they ask you to describe how DNA replicates, actually, the one you describe in details is the semi-conservative hypothesis. So, come with me in the next episode as we expound on the semi-conservative hypothesis in details. Thank you very much. May God bless you.